So now let's talk about, we'll go back to Mendel, and let's start talking about dihybrid crossing. This is when we're dealing with two traits, not just one. We've been talking about just one trait, but now, what if we talk about hair color and eye color? Then you're going to have much more of a mixture going on. But what he did was, and there's a lot of different traits you could do with peas, what he chose to do is that he wanted to see with pea seed color, not the petals now, the seed, and also the pea seed shape, whether they're smooth or wrinkled, okay, whether they're yellow or green. So we're putting another factor in there, okay? So we moved on to pursue more complex questions like inheritance of multiple traits. And he could have done all these different things. Pod colors, pod shape, seed colors, seed shape, flower colors, flower location, plant size. So many different traits, as much as we have a lot of traits. But he just wanted to just concentrate on the seed shape and the seed color. Okay? Whether it's smooth and or wrinkled or yellow and green. And you can have combinations of these four different things, which means, yes, it does get more involved. Okay? So he deduced that yellow color is dominating to green color. Okay? And smooth shape is dominating to the wrinkles. We'll just accept that now. If you want to learn more how he came up with that, just go back and probably read all about his studies that he did. But let's assume right now, to cut the chase, and just say what he found as his results. The yellow is the dominating factor, green is the recessive, smooth is the dominating, and wrinkled seeds are recessive. Okay, fair enough? Okay. So the symbols that he used, again, I'm not too fond of these symbols because they're capital, lowercase, but this is what he used, okay, is this. Capital S, lowercase s, capital Y, lowercase y. Okay? And he explained what dominant and recessives are. All right, so two trait cross between two true breeding varieties. All right, so these were the ones that are true breeding. One dominant for both traits. So, recessive for both traits. So what he did was, he had capital S, capital S, capital Y, capital Y. So smooth and yellow, both dominating, homozygous dominating. Okay? And he crossed it with the recessive ones. Lowercase s, lowercase s, lowercase y, and lowercase y. So you're crossing a wrinkled and green seed with a smooth and yellow seed. All right? You took the extremes. So there's no heterozygous here. Okay? You notice that the plants from this part, the dominating ones, homozygous dominating, they all produce SY, capital S, capital Y gametes. All right? And we also notice that this produced the lowercase s, lowercase s, the recessive, the homozygous recessive, produce it only the lowercase ones. All right? You do that enough, you know that they're true. Okay? Therefore, the first generation consisted solely of capital S, capital or lowercase s, capital Y, lowercase. They're heterozygous when you cross them over. Okay? So they all appear, the skins are smooth and yellow, but we know that they're heterozygous. Am I losing anybody? Just pay attention. You got to follow the letters. All right, so Mendel next allowed the first generation, F1 is first generation, individuals to self-fertilize. So you're going to do the heterozygous set with another heterozygous set. And what he found out, you want to memorize the numbers, but what he found out is ratios. He saw that smooth and yellow seeds were the majority of it. Then there was about equal amounts of wrinkled and yellow versus smooth and green. And then he saw a much lesser amount of wrinkled and green seeds. He did it a few times, and he got the same ratio for that. And the ratio was 9 to 3 to 3 to 1. So he saw that there was a pattern. He observed that. Statistics. You don't want to do this with only 4 plants, right? 4 seeds. You got to do, up to like 5, 6, 700 seeds. The bigger the sample size, the better your statistics come out. All right? So 2 trait crossings of other traits produce similar proportions of phenotypes. So that's what I'm saying. He did it not just one time, he did it a few times. And he got the same ratios. So this is what it physically, so if you want to actually see it instead of the numbers, that's what's going on over here. So he crossed the heterozygous over here and the heterozygous over here. Right? And he worked with the sperm and the egg versions of the plants, the carpels and the stamina, that kind of thing. And what he got was that. You can see what they actually look like. But it's a 9 to 3 to 3 to 1 ratio. All right? You see how this square, we were used to doing, well, it's not there anymore, but a 2 by 2 square. In order for you to work with 2 traits, you're going to have to now put a 4 by 4 square, give me 16 squares. It's more involved. The next test is going to be, pretty tight on this one. Okay? So again, this is just showing the same thing. All right? And there's the ratio showing numbers. So you can look at that if you want, but understand that it's the ratio there. So he came up with this law, Mendel's law of independent assortment. We also dealt with another Mendel law I talked about before about segregation. Here's his other law. And what it is that traits are inherited independently, not because of each other. In other words, just because you got blue eyes, that does not dictate what color hair you have. They work independently. Does that make sense? Okay? So each pair of alleles separate randomly and not controlled with the other pairs of alleles. We now see this. He didn't understand why, but 20 years later they started talking about chromosomes and meiosis. But now you understand the crossing over that's happening. But he's the one that was the spark in the bucket to get people thinking about this. Where in the cell does this all happen? And it's the meiosis that's happening here. Okay? So this is the reason for multiple traits. All right? The flipping of the coin, right? We're doing that in the lab. The result of one flip has no impact on the outcome of the next flip. That's basically what this is in terms of Mendel's law of independent assortment. Just because you flip the coin for blue eye color, the next time you flip it, this has no bearing on what you just flipped the first one for the second trait. Does that make sense? They're separate entities. Okay? So in the same way, alleles of one gene segregate into a gametes independently of each other's, or of another gene's alleles. In other words, when crossing over occurs, we don't know where that crossing over is going to occur. It does it randomly from one chromosome to another. Okay?
Have you ever been working on a Punnett square and you think, man, I really wish there were more than just these 4 squares? Well, if you ever have, today is your lucky day. And if not, well, prepare to have more squares. because up until this point we have focused on one pair of alleles, hence a monohybrid cross, where mono means one. In guinea pigs, for example, the trait of having or not having hair is influenced by a pair of alleles. But guinea pigs have more traits than just hair, right? Well, if you perform a cross with two pairs of alleles, that's called a dihybrid cross. The root di means two, so two pairs of alleles. One of us has this cat named Moo. Moo is in what the cow says, because he kind of looks like a cow, and we're not that creative with names. Anyway, the thing about Moo is that he is a cat that loves sinks. You get up in the morning and you try to splash water on your face, and oh no, there's a cat in your sink. And you try to brush your teeth, and no, there's a cat in your sink. And you have people over and you're terrified that they're going to flip out because there's a cat peeking out of the sink. Now, not all cats like sinks, but Moo does. Around the clock, chances are he's in the sink. We cannot find any information about whether this is a genetic trait. We don't know if Moo's parents liked sinks. But then we realize that Moo is not that weird because there's this website called catsinsinks.com. Seriously, Google it. You're going to find tons of cats in sinks. What if this was a genetic trait? Now, there's no research that we can find that demonstrates this is genetic, and it's probably not. Behaviors like that can be challenging to study. But for practicing dihybrid squares, let's imagine what it would be like if that was a genetic trait. So if this was genetic, hypothetically, cats that did like sinks would have to have a dominant allele, capital S, and cats that did not like sinks would have to have a pair of recessive alleles, lowercase s. Let's also take into account that cats, like guinea pigs in our previous video, typically have hair, but they can also be hairless. Having hair will be represented by the allele big H, and not having hair would require two recessive little h alleles. So let's say we want to cross a cat that is heterozygous for the trait of having hair and also for liking sinks. Heterozygous for both traits would be represented by the genotype big H, big S, little s. Now we want to cross that cat with a hairless cat that does not like sinks. To be hairless, the cat must be little h. All it takes is one dominant allele, a capital H in this case, for it to have hair. And if it does not like sinks, a recessive trait in our example, then it is little s. All it takes is one dominant allele, a capital S, and it would like sinks. So the second cat has a genotype of little s. Remember how it looks like in a regular monohybrid Punnett square? Here's an example of what it would look like if you were crossing a big H cat with a little h cat. Remember how you put the parents on the top and the sides like this? Well, when you are doing this, those alleles on the top and the sides, they represent the alleles that would be in the gametes of the parents. Gametes are sperm cells if male and egg cells if female, and they contain half of the genetic material as the cat's body cells. So it makes sense that if there are two alleles, the letters, in the big H parent, then a gamete would only carry one letter, a big H or a little h. This is known as Mendel's law of segregation. The gametes only carry one allele for a gene. Well, if you have a cat that is big H, big S, little s, then there are four alleles there. Two genes, one involving hair and one involving sinks. So if gametes carry only one allele, a letter, per gene, that means that each gamete is going to have two alleles. In those gametes, with each of them having two alleles, the letters, you have to account for every possible combination. Mendel's law of independent assortment says that those alleles are not linked. That means a cat can have hair and like sinks, or not have hair and like sinks. There is no link with the sink. It rhymes. So let's work out a dihybrid with the parent cross here. Step 1, write the parent cross with your 16 square Punnett square. Step 2, we have to get the gamete combinations from the parents. You know, what we write along the top and the side of the Punnett square. We really like the FOIL method to come up with these gamete combinations, but this is not the only way you can do this. We just like this way. So we're going to show you how to FOIL the first parent. And the first parent is big H, big S, little s. So to do FOIL, we do it like this. First, outside, inside, last. And you get these gamete combinations. Let's place those on the top of the Punnett square like this. Now let's go ahead and FOIL the other parent. The other parent is little h, little s, little s. So we'll do first, outside, inside, last. And you get these gamete combinations. Let's place those on the side of the Punnett square like this. And yes, they're all the same for this parent because notice that was all this parent could contribute as far as alleles. Now remember again, each gamete must have one allele, letter, of each gene. That's why you won't find a gamete with only H's or only S's. Remember, one allele of each. And now for step 3, combine the gametes to see what the offspring prediction will be. Now, for formatting purposes, because the parents had H's coming before S's, we're going to write it that way with the offspring as well. For formatting, you should also put capitals of each letter type before the lowercase. So in our example, what is the genotype ratio in the predicted offspring? Now remember that genotypes are the genetic makeups, the letters that represent the alleles. So 4 out of 16 ended up working for all these genotypes. That's a 1 to 1 to 1 to 1 ratio. It's kind of fun to say. Now, what about phenotypes? Well, half of the cats here have hair and half of the cats here don't. But with dihybrids, often you are asked about both traits. For example, what is the chance that there would be a kitten that has the same phenotype as our Moo? Moo has hair and he likes sinks. Well, it's a 4 out of 16, 25% chance that a kitten would have the same phenotype as Moo. We could write out that 4 out of 16 cats have hair and like sinks. 4 out of 16 cats have hair and dislike sinks. 4 out of 16 cats are hairless and like sinks. And 4 out of 16 cats are hairless and dislike sinks. This is also a 1 to 1 to 1 to 1 ratio. Now, some big things to remember. In our example, the genotype and phenotype ratios happen to be the same. This does not always happen. The handout will give you an example of when it's not. Just remember the steps to setting up the problem correctly and you'll be fine. Also remember that Punnett squares are predictions. This Punnett square is only predicting the chances of having offspring with certain genotypes or phenotypes. It's fascinating, really. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.